Hello everyone, welcome to today's live broadcast, the CytoFLEX S Innovative Benchtop Flow Cytometry for Research Applications. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter. At Beckman Coulter, we are dedicated to advancing and optimizing the laboratory. For more than 80 years, we have been a trusted partner for laboratory professionals helping to advance scientific research and patient care. We have a vital role. Our focus on innovation, reliability, and efficiency has led us to become the partner of choice for clinical research and industrial customers around the globe. For more information, please visit beckman.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, please click on the support button at the top right of your presentation window and submit your problem through the green Q&A button lower left. I would like to introduce today's speakers, Ala Darwish and Azam Ghauri. Ala holds a BSc in the pharmaceutical sciences. Throughout his career, he has worked with several blue-chip pharmaceutical and medical devices manufacturers. Allah aspires to elevate the healthcare provisions and life science research standards across the Middle East, Turkey, Africa, and is highly interested in understanding customers' and stakeholder-specific operational needs, hence tailoring customers' focused solutions and processes. Currently, he manages the Middle East and across the life science companies and Danaher. Azam earned his MSc in Bioscience Technology from the University of York. He worked as a field application specialist responsible for QIAGEN products with a local distributor in the United Arab Emirates. He then joined Eppendorf as a product specialist for the Middle East and Africa. Currently, he works with Beckman Coulter to support workflow cytometry in the Middle East, North Africa, and the Pakistan region. I will now turn it over to Ala for his short presentation where he introduces Beckman Coulter Life Sciences, which is part of the Danaher Life Science Group of companies.
Thank you for the nice introduction. I'd like also to thank you all for joining our webinar today. This is actually among a series of webinars for the Life Science Group at Danaher. At Danaher, customers talk, we listen. Quality is our focus so that we innovate products to you at a cost-effective parameter. We work together as teams to develop solutions that answers the questions that you raise to us at Danaher. Specifically for the Middle East, we have a team of application as well as technical support specialists who are based across the region, mainly in Dubai as well as in Saudi. The life science operating companies in Danaher are mainly 4 companies. Leica Microsystems, which serves different sectors, CyTExpert, Molecular Devices, and Beckman Coulter. For the purpose of our presentation today, we are going to be focusing on Beckman Life Sciences, which actually has more than 2,000 associates employed across the world, mainly in 49 offices. And we serve customers in academia, pharma and biotech research, as well as in hospitals. I would like to thank you again for joining my presentation, and I would like to hand the presentation again to the moderator.
Thank you so very much, Ala. I will now turn it over to Azam for his presentation.
Thank you, Robert. Good morning and good afternoon, everyone. I'm excited to talk today about the CytoFLEX S flow cytometers, which are an extension of the innovative CytoFLEX platform from Beckman Coulter Life Sciences. So let's begin. The original CytoFLEX system features a compact footprint, integrated detection optics and lasers, and a simplified but highly reliable fluidic system. It is available in 21 standard configurations with up to 3 lasers and 13 fluorescent detectors to provide the great application flexibility. The CytoFLEX S expands your research possibilities with a 4th laser option. It continues to deliver superior performance with the ease of installation and operation for research application the CytoFLEX is known for. Let's talk about the configurations available with the CytoFLEX S. So the CytoFLEX S system is available in preset configurations with up to 4 lasers from a choice of 5. While the CytoFLEX can be had with blue 488, red 638, and violet 405 nanometer lasers, the CytoFLEX S adds the option of yellow-green 561 nanometer laser and the near UV 375 nanometer laser. So these lasers are spatially separated and powerful. To give you an idea, the violet laser available on the CytoFLEX and CytoFLEX S is 80 milliwatt. The near UV laser comes with 60 milliwatt, while the standard blue and red lasers are 50 milliwatt, and the yellow-green laser is 30 milliwatt. When you have the blue laser, you have the option of exciting common dyes like FITC, PE or phycoerythrin, ECD from Beckman Coulter, PC 5.5 or PERCP, and TC7. With the red lasers, you have the option of exciting APC, APC ElixirFluor 700, and APC ElixirFluor 750. The violet laser offers you up to 5 channels in which you can have Pacific Blue, Chrome Orange from Beckman Coulter, and the Brilliant Violet 605, 650 dyes, tandem dyes. The yellow-green laser gives you the option of exciting PE and PE tandems along with fluorescent protein. So the CytoFLEX systems will easily fit on the bench or in the biohood. The CytoFLEX weighs about 24 kilograms without the plate loader, less than 50 pounds, and it can be easily relocated by the technologist. The single tube loader of the CytoFLEX and CytoFLEX S supports the standard 5 ml flow cytometry tubes, which are 12 by 75 millimeter in dimensions, but you can also use 1.5 and 2 ml microcentrifuge tubes. Also available is an optional 96 well plate loader that can be ordered as factory installed or as an upgrade for CytoFLEX in the field. Adding the plate loader does not change the dimensions of the CytoFLEX, and the operator has the choice of switching between the plate loader or the single tube loader modes in less than 5 minutes. So the plate loader accepts your standard U-bottom, V-bottom, and flat bottom 96 well plates. The advantage of plate loader is, of course, that you can use less sample volume. You have options for mixing, and these plates can be ordered from Beckman Coulter or the generic options available from other vendors. In this slide, we talk about the performance. The secret behind the CytoFLEX high performance is the innovative Wavelength Division Multiplexing technology, or WDM, that we use with avalanche photodiodes. How does this work? Fluorescence and scatter light signals are directed to the WDM module via optical fibers. The light is then passed through a series of optics and filters which direct and focus specific wavelengths onto an avalanche photodiode detector. This avalanche photodiode detector replaces the PMTs used in conventional flow cytometers. APTs have higher quantum efficiency compared to PMTs, and they have better efficient performance in the far red light spectrum for APC dyes. If you spec the CytoFLEX with the violet laser, you also have the option of having side scatter collected from violet laser illumination. 405 nanometer laser light causes greater side scatter signals than 488 nanometer laser light. So when you use violet side scatter, it is a great feature perfect for looking at exosomes and microparticles to as small as 200 nanometer in size. In this slide, we show the use of Spherotech 8-peak beads on a standard CytoFLEX system with blue, red, and violet lasers. What I would like to highlight is that conventionally, flow cytometers struggle when it comes to resolving the dim particles or the dim beads in the far red channels of PC7 and APC. This is no longer an issue with the CytoFLEX, which utilizes the WDM technology with avalanche photodiodes. You can see clearly that the rainbow beads are well resolved, and you can see all the 8 peaks in the PC7 and APC channel. When it comes to standard channels for FITC and PE, you can see that the sensitivity of the CytoFLEX is class leading. Now let's talk about the utility provided by the 561 nanometer yellow laser. The 561 nanometer yellow laser is perfect for exciting fluorescent proteins like GFP, DS-Red, D-tomato, and mCherry. Here you can see a comparison between a standard blue laser CytoFLEX system versus a CytoFLEX S equipped with the 561 nanometer laser. When using 6-peak mCherry beads, you can see that you can resolve all the 6 peaks using the yellow-green laser. When using bacteria labeled with mCherry, the signal-to-noise ratio, or the separation between the positive and negative peak using the 561 nanometer laser, is much superior to blue laser light illumination. Now let's talk about the near UV 375 nanometer laser option available on the CytoFLEX S. Using the near UV laser allows you to excite brilliant ultraviolet reagents without incurring the cost of a true UV laser. In this example, you can see whole blood stained with DAPI and CD3. The CD3 is labeled with brilliant violet 737 and Hoescht dye. You can use the brilliant violet 737 with a 780 bandpass filter or 712 nanometer bandpass filter. You would want to avoid using 712 nanometer bandpass filter if you are going to use dyes like Alexa Fluor 700 on red laser line. Let's go to another slide. Here we are looking at the 12-color experiment, which includes brilliant ultraviolet dyes and standard dyes used on the blue and violet laser lines. This is an immunophenotyping application where we are looking at CD14 positive monocytes, lymphocytes labeled with CD3, which are T lymphocytes, CD56 and 16 labeled natural killer cells, and the separation between CD4 and CD8 T cells. If you look at the performance of CD3, which is labeled with brilliant ultraviolet dye, you can see that the resolution between the positive and negative population is clear to see. In this slide, we are looking at cell viability using Hoescht and DAPI. DAPI can be excited by the violet or the near UV laser, and you can see the results are comparable with Hoescht, which is excited by the near UV laser. So again, you can use the near UV laser with 80 milliwatt power on the CytoFLEX S instead of using a much more expensive and heat-generating ultraviolet laser. So I talked about the hardware specifications and features. Let's come back to the software available with the CytoFLEX. The CytoFLEX and CytoFLEX S, no matter the version, comes with CytExpert software. CytExpert software is easy to learn, easy to use, and easy to maintain. It supports compensation using cells or beads. It supports quality control using CytoFLEX daily quality control beads. And we also provide training for the CytExpert on our website free of charge. This is a fully featured software which has acquisition and analysis modes. If you're familiar with the DIVA software, the CytExpert software is easy to pick up and use. So we provide complete instrument quality control using the CytoFLEX daily QC beads. The target channel settings are provided on the web, which are imported into the CytExpert software, which automatically does QC, checks the fluidics and the alignment, calculates the laser delay, and lets you know if any parameter has failed and why. The quality control results are tracked using Levi-Jennings graphs, automatically saved in the quality control module of the CytoFLEX QC. Let's talk about compensation and how CytoFLEX brings innovation even to this challenging aspect of flow cytometry. One thing which we have to emphasize, and which is an advantage of the avalanche photodiodes compared to PMTs, is that the APDs, the photodiodes, are much more linear compared to PMTs. We do not adjust the voltages in the CytoFLEX. We just increase the gain. And because of the linearity, when you adjust the gain, the software is actually able to calculate new compensation factors on the new gain. What this allows you to do is to run one experiment with positive controls with the dyes and store in a compensation library in the CytExpert software. If for a different experiment you change the gain settings, in other words, you change the sensitivity of the photodiodes, you do not have to run the positive controls again to calculate the new compensation matrix. The software is able to adjust to the new gain settings and transform the compensation matrix. This makes it much easier and saves you time. In this slide, I talk about the analysis features available with the CytExpert software. The CytExpert software has a fully featured analysis suite with all the ability to show the different plot types you would like to use in analysis, including overlays. It has ease of use features like dragging and dropping plots anywhere in the workspace, panning inside a plot, zooming into a population of interest, and plots to show the gating hierarchy and colors of the populations of interest. The CytExpert software can be used offline. It requires a computer with Windows 7. It supports Windows 7 32 and 64-bit. It is provided free of charge with the instrument. It can also be copied over a USB drive and installed on your personal computer, as it does not require a license or a dongle to be used. The files generated by the CytExpert software utilize the FCS 3.0 standard, and you can export the flow cytometry files from this software, which can be read by other analysis software like Kaluza from Beckman Coulter, FCS Express, and Flojo. Let's talk about the maintenance side of things. The CytoFLEX is designed to be simple to use and to live with in your lab. You have to purchase a limited number of consumables. There is the sheet fluid, like any conventional flow cytometer. You have Contrast 70, which is used for deep cleaning. You have Flow Clean, which is a detergent-based cleaner to get rid of proteins left in the tubing. And then you have an easily replaceable sheet filter, which is disposable and designed to be replaced by the operator himself in 5 to 10 minutes every 6 months. The CytExpert software aids you in the maintenance by having pre-built startup and initialization cycles. It can backflush automatically between samples to minimize sample carryover. It has priming functions, and it has shutdown cycles, which utilize just three reagents. So to summarize, the CytoFLEX platform, of which CytoFLEX S is an extension, gives you a high-performance system in a compact system. This performance is enabled due to the use of the innovative WDM module with avalanche photodiodes. And the choice of lasers and fluorescence detectors allows you to choose the CytoFLEX system which is suitable for your current research project. Not to forget, the CytoFLEX and the CytoFLEX S are upgradable in the field as your research fields and requirements expand. It comes with software which is fully featured, intuitive, and the maintenance is simple of the system. With this, I conclude my presentation, and I would be excited to receive questions at this stage. Over to you, Robert.
Thank you so very much. And thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We're going to try to answer as many questions as time allows. Our first question, can CytoFLEX S be upgraded to support more colors?
Yes, the CytoFLEX S has upgrade pathways available. You can have a system which can have blue, yellow, violet lasers, or you can have a system which has blue, yellow, red, and near UV capabilities. However, we can start with a CytoFLEX S system which has 6 fluorescent detectors unlocked, and you can go up to unlocking all the 13 fluorescence detectors when your application needs to require it and you have the budget to match. The only limitation in the upgrade pathway is that if you have ordered a system which has 4 lasers, excluding the 5th laser, which can be near UV or yellow or violet, you cannot have the 5th laser, which was excluded in the first place, into the CytoFLEX S system you have. So to conclude, it is easy to unlock fluorescent detectors in the CytoFLEX S, but if you already have 4 lasers, you cannot add the 5th laser later on. I hope this answers the question.
And our next question is the CytExpert software easy to learn?
Yes, we definitely believe so. And the proof is in the pudding that you can go to our website and get online training using on-demand web-based computer training program, which will also issue you with a certificate. The CytExpert software we find, as per feedback from field application specialists, can be taught in 1 or 2 sessions. Thank you.
Thank you. Our next question, can we expect more colors and or more lasers in the future?
At the moment, we have no options of increasing the number of colors available on the CytoFLEX S platform. However, we will be adding a 6th laser option for the CytoFLEX S, which will be in the infrared spectrum to excite dyes like ElectroFluor 780. So pretty soon, you will have the option of choosing 4 lasers for CytoFLEX S from a choice of 6 lasers. Thank you.
It supports FCS 3.0. So by default, it supports old versions of SCS files?
No, I'm afraid not. The FCS 2.0 and 2.1 files from the older generation of flow cytometers is not supported by the CytExpert software. Thank you.
There are multiple configurations available. Any recommendations on choosing a starter configuration that can be upgraded in the future?
Right. So this is an interesting question. You have to decide in the beginning which are the fluorochromes you're going to use. If your work is going to focus more on using fluorescent proteins, then you should choose a CytoFLEX S system with the yellow laser. If you're going to work with DNA staining, stem cell site populations, then you would go for the near UV laser. When it comes to the other laser choices, you have to choose which is more important, having the blue and red laser combination for the very common fluorochromes or having a blue and violet laser combination. The violet laser is especially useful if you're going to look at nanoparticles. So, like I said, as for my answer for a previous question, it is quite possible to upgrade the capabilities from 6 to 8 to 13 colors, but you have to choose the correct 4 lasers from the start. If you exclude a laser, it cannot be added in the future. Thank you.
And our next question, could you give the speed of the sheet or sampling flow?
Yes, I've loaded up a slide to show this. We have 3 preset sample rates. Slow, which will take up 10 microliter per minute, medium, 30 microliter per minute, and fast, which will take 60 microliter per minute. However, you also have a custom sample flow rate slider in which you can go all the way up to 240 microliter per minute. As for sheet consumption, the CytoFLEX has the option of putting it on standby mode. When the system is on standby mode, it will not be running sheet fluid through the flow cell and the Fluidics. And only when you need to run a sample, you will initialize the system and consume the sheet according to your preset sample rates. I hope this answers the question. Thank you.
And thank you. Allah and Azam, do you have any final words for the presentation for today?
Yes, I would like to say that the CytoFLEX platform is something which Beckman Coulter is very actively working on. We came up with the original CytoFLEX and globally released it in 2015. Soon after, we added the plate loader option. In 2015, from the last quarter, we introduced the CytoFLEX S with the yellow-green laser. And this year, we have added the near UV laser option along with additional band pass filters for the yellow laser system. Like I mentioned, we are going to add a 6 laser option, and we are also listening to the customer feedback, and we will be open to developing a extended CytoFLEX which will have more than 4 lasers at the same time. I hope this answers the question. Thank you.
Thank you. And again, thank you to Allah and Azam for their outstanding presentation. I would also like to say a special thank you for Beckman Coulter for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through October of 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Until next time, goodbye.